Hi friends, welcome to CyberC. In this video, we will be dealing with early experiments and light reaction which comes under photosynthesis in higher plants. Photosynthesis is a physico-chemical process by which light energy is used to drive the synthesis of organic compounds. Important due to two reasons: primary source of food on earth and release of oxygen into the atmosphere by green plants. In 1770, Joseph Priestley performed a series of experiments that revealed the essential role of air in the growth of green plants. He discovered oxygen in 1774. He observed that a candle burning in a closed space like a bell jar soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized that the plants restored to the air whatever breathing animals and burning candles remove. Jan Ingenhouse showed that sunlight is essential for photosynthesis. In an elegant experiment with an aquatic plant, he showed that in bright sunlight, small bubbles were formed around the green parts, while in the dark they did not. Later, he identified these bubbles to be of oxygen. Hence, he showed that it is only the green part of the plants that could release oxygen. In 1854, Julius von Sachs provided evidence for production of glucose when plants grow. His studies showed that the green substance in plants, chlorophyll as we know it now, is located in special bodies, later called chloroplasts, within plant cells. He found that the green parts in plants is where glucose is made and that the glucose is usually stored as starch. T. W. Engelmann, using a prism, split light into its spectral components and then illuminated a green alga, Cladophora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of O2 evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and B. Here, those with the red color represents the bacteria, aerobic bacteria, and the green is the algae cladophora. In 1930s, Cornelius van Niel, a microbiologist, observed photosynthesis in purple sulfur bacteria. He observed that CO2 plus 2H2S plus light energy gave CH2O, a carbohydrate product, plus H2O plus 2S. Here, H2O is not the hydrogen donor, whereas H2S instead is the hydrogen donor and the oxidation product is sulfur or sulfate. So he demonstrated that photosynthesis is essentially a light dependent reaction in which a hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. In green plants, H2O is the hydrogen donor and is oxidized to O2. Within the chloroplast, there is the membranous system consisting of grana, stroma lamellae, and the fluid stroma. The membrane system is responsible for trapping the light energy and for the synthesis of ATP and NADPH. It includes light absorption, water splitting, oxygen release, and formation of ATP and NADPH. These reactions are directly light driven and are called light reactions. Later, the products of light reactions, namely ATP and NADPH, are utilized in dark reactions which occur within the stroma of chloroplast to incorporate CO2 and for the synthesis of sugar. In the thylakoid membrane, The pigments are organized into two discrete photochemical light harvesting complexes, PS1 and PS2, photosystem 1 and photosystem 2. They are named in the sequence of their discovery. All the pigment molecules except a single molecule of chlorophyll A form antennae. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. Antennae plus reaction center is equal to photosystem or light harvesting complex. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer, hence is called P700, while in PS2 it has absorption maxima at 680 nanometer and is called P680. From the diagram, it is visible that the photons which are absorbed by the pigment molecules are transferred to the reaction center, and from the reaction center the electrons which are excited due to the energy from the photon are received by the primary acceptor. So what happens during the light reaction? The light with the wavelength 680 nanometer has the ability to excite electrons from photosystem 2. So the electron excited from photosystem 2 is received by an electron acceptor with a higher redox potential. Hence the electron is moved uphill, and from there through an electron transport system which includes B6F complex and plastocyanin, it is transported to the PS1, photosystem 1. The electrons from photosystem 1 can be excited only by a light of wavelength 700 nanometer, and from there the electrons are moved or transported to electron acceptor, or they are received by an electron acceptor which is also having a higher redox potential, and finally these electrons are utilized for the production of NADPH. So this shape is formed only when all the carriers are placed in a sequence on a redox potential scale. The whole scheme of transfer of electrons, starting from PS2 uphill to the acceptor, down the electron transport chain to PS1, excitation of electrons transferred to another acceptor, and finally downhill to NADP plus, causing it to be reduced to NADPH and H plus, is called the set scheme. It is called due to its characteristic shape, and this shape is achieved only when these carriers are placed in the sequence of a redox potential scale. So this is what is happening during the light reaction, and the primary electron acceptor which accepts electron from the PS2 is plastoquinone, a small molecule. Okay, it further mediates the electron transport system, and the water splitting complex is associated with PS2, and it is physically located on the inner side of the membrane of the thylakoid. So splitting of water is also occurring during this light reaction. What is the difference between cyclic and non-cyclic photophosphorylation? During cyclic photophosphorylation, only photosystem 1 is functional, photosystem 2 is non-functional. This mainly occurs within the stroma lamellae membranes because they lack photosystem 2. Also, they lack NADP reductase enzyme. Hence, in the cyclic flow of electrons, that is during the cyclic photophosphorylation, NADPH is not synthesized, only ATP synthesis take place. Okay. Non-cyclic photophosphorylation is the set scheme that we just mentioned or discussed before. Now about the chemiosmotic hypothesis. According to this hypothesis, a proton gradient is created across the thylakoid membrane, which is later utilized for the synthesis of ATP. How this proton gradient is created? There are mainly three reasons why this gradient is created. The first reason is that the splitting of water, which is associated with PS2, releases H plus into the lumen. Hence, the H plus is being accumulated in the lumen. As electrons move through the photosystem, the protons are transported across the membrane. That is the second reason. Because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an H plus carrier. So this molecule removes a proton from the stroma, and while transporting an electron to the electron carrier, next to an electron carrier on the inner side of the membrane, it releases the proton also along with that into the inner side. Okay, so in that manner H plus is transported from the stroma towards the lumen, By means of this, electron transport system molecules. Now, the third reason is that the NADP reductase enzyme, which is located on the stroma side of the membrane, utilize H plus for reducing NADP plus to NADPH. Okay, so in this manner, protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen there is accumulation of proton. This creates a proton gradient across the thylakoid membrane. So this gradient is later broken down due to the movement of protons across the membrane back to the stroma through the transmembrane channel of the F0 of the ATPase. The ATPase enzyme, we can see in the diagram, consists of two parts. F0 is the one which is embedded in the membrane and forms a transmembrane channel that carries out the facilitated diffusion of protons across the membrane. The other portion is called F1 and protrudes on the outer surface of the thylakoid membrane that faces the stroma. The breakdown of the gradient provides enough energy to cause the conformational change in the F1 particle. So conformational change is occurring within the F1 particle and the transfer of electrons is occurring through the F0 particle. So this makes the enzyme synthesize several molecules of energy packed ATP. So yes, that's it. I hope you are all clear with the light reaction. The ATP and NADPH that is produced during the light reactions will be utilized in the dark reaction for producing sugar. We will be dealing with the dark reaction in our next video. For now, thank you all. Please like, share, subscribe and support this channel.